This is the last lecture on Chapter 7. So we'll be looking at a variety of different types of neurotransmitters. One of the major types of neurotransmitters are monoamines. And these can be broken down into the largest group, which is catecholamines. These are derived from amino acid tyrosine. This includes dopamine, which is a neurotransmitter, norepinephrine, and epinephrine. Norepinephrine and epinephrine are also both neurotransmitters. They're also pretty well known as being hormones that are released by the adrenal glands. Serotonin is another very important neurotransmitter. It's involved in things like well-being, maternal behavior, has a variety of different effects in the brain. It's a little bit different. It's derived from tryptophan rather than tyrosine. And histamine is another neurotransmitter. It's derived from histidine. Now, histamine is pretty well known also in the fact that it's involved in the inflammation. So antihistamines, for example, reduce inflammation of the skin. Previously, we used acetylcholine, or ACH, as our example of a neurotransmitter. Monoamines are very similar in the sense that they're made in the presynaptic axon terminal, or synaptic bouton. They're released via exocytosis when a synaptic vesicle containing monoamines fuses with the plasma membrane. The monoamines then diffuse across the synaptic cleft to the postsynaptic cell where they bind to specific receptors. They're quickly taken back into the presynaptic cell, and this is called reuptake. And there they're degraded by an enzyme called monoamine oxidase, MAO. This is an illustration from your book indicating some of the things I've just talked about. You have monoamines that are produced and stored in synaptic vesicles. So you start with, for example, tyrosine that can be converted to dopa. That's a precursor of dopamine. Dopamine can be converted to norepinephrine. And then this would be stored in a synaptic vesicle. The synaptic vesicles can then fuse with the membrane. The vesicular membrane now becomes part of the plasma membrane. At the same time, the neurotransmitter is released. It moves by simple diffusion across the synaptic cleft and then binds to postsynaptic receptors, thereby having an effect on the postsynaptic cell. In this case, it's norepinephrine. Now, in this case, with norepinephrine, most of this neurotransmitter is their reuptake mechanisms, proteins that will bring the norepinephrine back in. And then once it's back in the cell, it will be inactivated by by monoamine oxidase. And then you can go back through the whole process of resynthesizing the neurotransmitters you need. And the way the neurotransmitter release occurs is very similar to what we talked about with ACH. So an action potential will invade the axon terminal or synaptic bouton. There will be an influx of calcium through voltage-gated calcium channels. This calcium will then interact with the protein synaptotagmin, which then interacts with the snare complex of proteins, which help these vesicles dock with the plasma membrane. And that'll then lead to the fusion of the vesicular membrane with the plasma membrane. Recall that previously when we talked about acetylcholine, there were two types of postsynaptic receptors. There were nicotinic acetylcholine receptors, and those were typically ligand-gated, meaning ion channels, meaning that the two molecules of acetylcholine would bind to the postsynaptic receptor protein. The receptor protein itself was a channel. It would open up and it was equally permeable to both sodium and potassium. And there was a net, because of a difference in driving force, there was a net influx of sodium that was greater than the net efflux of potassium. Therefore, nicotinic acetylcholine receptors are excitatory. They depolarize the postsynaptic membrane. Now, for muscarinic receptors, now those are typically G protein coupled receptors, which we talked about, where the acetylcholine will bind to a G protein coupled receptor. The G protein is activated, and that may lead to other types of postsynaptic effects, such as the opening of a potassium channel. And so, or it could be other effects like activation of adenylyl adenylate cyclase, which would lead to an increase in cyclic AMP in the postsynaptic cell. Now, for monoamines, none of the receptors of these signals are direct ion channels. They all use a second messenger system. So an example would be, again, we could use cyclic AMP as being the most common second messenger for catecholamines. In the next slide, I'll show a picture of this, but let's just go over the basic idea. So with your typical monoamine, you'll have binding of a catecholamine to its receptor. So this is going to be a G protein coupled receptor. That's going to activate a G protein. It dissociates, and will typically you might have, for example, the alpha subunit will translocate laterally in the membrane. It will bind to an enzyme called adenylate cyclase. This is also called adenylyl cyclase. Those are the same thing, which converts adenosine triphosphate ATP to cyclic AMP. So the cyclic AMP is now the second messenger. The first messenger was the neurotransmitter. Cyclic AMP activates an enzyme within the cytosol called protein kinase, which then will phosphorylate other proteins. And these other proteins could, for example, be, in fact, ion channels. So it could, for example, lead to the opening of an ion channel, or the other proteins within the cell could be, for example, other enzymes or things like that. Okay, this slide illustrates what happened with norepinephrine. So norepinephrine will bind to its receptor. This binding causes the activation of a G protein, which has three subunits, alpha, beta, and gamma. Now, the alpha subunit, once it's activated, the alpha subunit may translocate laterally in the membrane. It's also potentially possible for beta and gamma to also translocate and also have some sort of action within the membrane when it binds to another protein. In this case, we're showing the alpha binding to adenylate cyclase. Now, this is an enzyme. It converts ATP to cyclic AMP, which is a second messenger. Cyclic AMP can then interact with a protein kinase, which is inactive, making it active. Now, recall what a protein kinase is. It's a protein that will transfer inorganic phosphate, typically from ATP to another molecule, phosphorylating it. So it could phosphorylate other proteins, which might be enzymes within the cytoplasm. It could also phosphorylate ion channels, thereby either opening or closing the ion channel and having a resulting effect on the membrane potential of the cell. Another very important neurotransmitter is serotonin. One place your body uses serotonin is in the brainstem. There's a region called the raphe that lies right at the midline of the brainstem. Serotonergic neurons there release serotonin onto neurons that are involved in respiration. In other words, these are neurons that generate the respiratory rhythm for breathing. So those are very important. Serotonin is also implicated in mood. Generally, if you have more serotonin, you feel better. It's involved in various behaviors, such as maternal behavior. In mice, for example, a higher level of serotonin correlates to higher levels of maternal care. Appetite, so it increases appetite, and it can also increase cerebral circulation. The drug LSD is an agonist, so it acts on serotonergic receptors. Serotonin is very important, as I said, for mood. And so for people that are depressed, one treatment is to use a serotonin-specific reuptake inhibitor, SSRI. What these do is they prevent the reuptake of serotonin back into the presynaptic terminal. Therefore, the serotonin is left in the synaptic cleft for longer, and therefore it can activate the postsynaptic receptors. And this allows for more activation of the postsynaptic cell, and this leads to a better mood. There are over a dozen different receptors for serotonin, so different drugs will target specific serotonin receptors. So, for example, some will target anxiety, so it will be anxiolytic, relieving people's anxiety. For certain obese patients, serotonin can help with appetite control, and serotonin can help with migraine headaches in other patients. Dopamine is another very important neurotransmitter. You have dopaminergic neurons in two areas of the midbrain. First of all, you have nigrostriatal dopamine neurons. These are involved in motor control. Those start in areas such as the substantia nigra, and they go to a striated region of the brain called the dorsal striatum. So this is involved in motor control. And there's the mesolimbic dopamine system. This is involved in emotional reward. So these neurons start in the VTA, or ventral tegmental area, and they go to the forebrain, regions like the amygdala, which are involved in emotion, olfactorily tubical nucleus accumbens, which is involved in reward, prefrontal cortex, neocortex. So this is involved in executive functions, such as planning and things like that. Let's look at a little more detail at the nigrostriatal dopamine system. So these neurons start in the substantia nigra. This is part of what are called the basal nuclei. Nuclei, again, are groups of cell bodies that have a similar function and are grouped together within the central nervous system. So they're the equivalent of a ganglion, but within the nervous system. So these send processes to the corpus striatum, shown here. So this is very important in control and initiation of movements. So in Parkinson's disease, there's a degeneration of these neurons, of the nigrostriatal dopamine system. So to treat them, there's a couple ways you can treat them. One is to give them L-DOPA. L-DOPA is a precursor of dopamine. So even though they have fewer neurons, they're able to generate more dopamine neurotransmitter, which is released. And another way to treat these patients is with monoamine oxidase inhibitors. So recall that monoamine oxidase breaks down the dopamine that's been released when it's taken back up into the presynaptic terminal. So if you inhibit that process, that can lead to more dopamine being available in the synaptic cleft, and therefore more activation of dopaminergic synapses. The mesolimbic dopamine system starts in a region of the midbrain, such as the ventral tegmental area. It sends processes that are involved in emotional reward. And these systems are also associated with addiction, such as nicotine addiction, alcohol addiction, and other drugs. And as I mentioned before, a lot of that goes through the nucleus accumbens, which is thought to be a center that's involved in reward. In other words, typically, if you do something like you work hard, then you get to make money, and then you can have a nice meal and have a nice place to live and so forth. But if you activate this pathway by taking drugs, you sort of get the reward without the hard work. And so this pathway gets activated through alcohol, nicotine, and other drugs. So in a sense, it short circuits the way the system's supposed to work. Schizophrenia is associated with too much dopamine in the system. And there are drugs that treat schizophrenia that are dopamine antagonists. Another very important neurotransmitter is norepinephrine. This is used both in the central nervous system and the peripheral nervous system. Sympathetic neurons in the PNS use norepinephrine on smooth muscles, cardiac muscles, and glands. So recall that the sympathetic division of the autonomic nervous system is involved in the fight or flight response. So this system is activated when you're scared, when you're angry. Also during exercise, that's a very good way to increase sympathetic output. So during that time, norepinephrine acting on smooth muscles will tend to essentially reduce the activity of smooth muscles. It will increase the activity of cardiac muscles. So for example, with the heart, it will act on pacemaker cells in the sinoatrial node, which is in the right atrium, and that will increase the heart rate. Sympathetic neurons can also act on glands within the body. Norepinephrine is used by neurons in the CNS. It's often thought to be associated with arousal. So, for example, there are certain drugs like amphetamines that will stimulate these norepinephrine pathways in the brain, and those tend to lead to greater arousal, in terms of being, more awake, more activated. There are a host of other neurotransmitters that are very important. Among these important neurotransmitters, there's glutamate. This is an amino acid that's used as a major excitatory neurotransmitter in the brain. So if you look in your brain in the cerebral cortex, the majority of the EPSPs produced at synapses are in fact due to glutamate being released at presynaptic terminals. And your book points out a really interesting fact, which is that energy required for all the EPSPs constitutes the major energy use of the brain. So apparently there's more flux of ions during EPSPs and other synaptic inputs than during action potentials. Astrocytes take up glutamate from the synaptic cleft. So we did talk about this before. At a presynaptic terminal, you have release of glutamate from vesicles. It then diffuses into the synaptic cleft and across the postsynaptic cell where it will bind with receptors. Now, eventually, you can't have the synapse being activated forever. So one way you can get rid of the glutamate is for astrocytes to essentially uptake the glutamate. This reduces then the concentration of glutamate in the synaptic cleft. Astrocytes can also increase glucose uptake and increase blood flow by vasodilation in active regions. So again, going back to this idea of there being a large energy cost for these EPSPs, you would want more glucose uptake in a region of the brain that's very active, and you'd also want more blood flow to bring the glucose to those active regions. There are different types of glutamate receptors. All of these serve as ion channels. So there's the NMDA channel, what's a channel receptor. There's AMPA receptors and kainate receptors. And I'm not going to really go into great detail talking about these. Just be aware that there's different types of receptors. And again, I'm not going to go into the details because this is a sort of a whole portion of a lecture on its own. But the NMDA and AMPA receptors are very important for the formation of memories, so learning, and they're also the next step, which is memory storage. So they're involved in both those processes. So far, we talked about neurotransmitters that generally have an excitatory effect. However, it's necessary to have inhibitory neurotransmitters also. So glycine is an amino acid. It's used as a neurotransmitter. It produces inhibitory postsynaptic potentials, or IPSPs. Binding of glycine opens chloride channels, causing an influx of chloride. There's a higher concentration of chloride outside than inside, and that's due to the active transport of chloride across the plasma membrane of a neuron. Having said that, the equilibrium potential for chloride is actually very close to the resting membrane potential of a cell. So the resting membrane potential of a cell is, say, -70. The chloride equilibrium potential may be -70, or slightly below that, like, say, -80. Okay. Activation of these chloride channels makes it harder for the membrane to reach spike threshold, so they have an inhibitory effect. So in other words, I've discussed this previously in a previous lecture that an IPSP is defined as a synaptic event that reduces the probability of a spike occurring. And so if you bring the membrane further away from the spike threshold, you reduce that probability. Glycine is important in the spinal cord for regulating skeletal muscle movement. This allows antagonist muscle groups to relax while others are contracting. So a good example of this would be your biceps and your triceps. Those are antagonistic muscles. So for example, if you're flexing at the elbow, so you're flexing the forearm, you'd have activation of the biceps. Now, simultaneously, you'd like to not have the triceps contract. So you should have relaxation of the triceps. So the motor neurons that innervate the triceps, in other words, the somatic motor neurons that go to the triceps, should not be generating a high number of action potential, a high frequency of action potential. In fact, they could be silent or just have a low frequency. And glycine is very important for that. So glycine is released from interneurons that synapse with the motor neurons that go to the triceps. So while the biceps motor neurons are active. The tricep motor neurons are being inhibited due to inhibition they're receiving through glycinergic synapses from interneurons. As I mentioned before, most neurons many of the central neurons, in other words, ones in your brain or in your spinal cord, generally going to receive a combination of EPSPs and IPSPs. In other words, they're getting excitatory synaptic input as well as inhibitory synaptic input. And this is true for the motor neurons that control the diaphragm. The diaphragm is the major muscle that's involved in breathing. So, for example, when the diaphragm contracts, it opens up the thoracic cavity, increasing lung volume so that you will breathe in. The poison strychnines blocks glycinergic receptors, and this leads to death by asphyxiation. The reason is these motor neurons that go to the diaphragm simply become too excited all the time. So they essentially just contract. But they stay contracted, they don't relax. You're not able to breathe in and out. You just breathe in, and then you continue to breathe in, essentially, but in the sense that the diaphragm is contracted. So, of course, at that point, there's no more further movement of air into the lungs, and you essentially die by not being able to breathe. And so this is why strychnine is used as, for example, a poison for mice and rats. It kills them by asphyxiation. And it also is very effective at killing people. So it's ended up being used in lots of like murder mystery type books. Okay, so glycine is extremely important in the central nervous system as well as in the peripheral nervous system. Another very important neurotransmitter is GABA, gamma aminobutyric acid. It's the most common neurotransmitter in the brain. It's used at about a third of the brain's neurons, by 1/3 of the brain's neurons. It's generally going to have it always has an inhibitory effect. So it is released postsynaptically. Chloride channels were open when it binds to the receptor channels. The chloride will flux from outside to inside, therefore hyperpolarizing the postsynaptic neuron. So it's inhibitory. So these are IPSPs that are generated by GABAergic synapses. GABA is involved in motor control, and so degeneration of GABA-secreting neurons in cerebellum. So the cerebellum is not the cerebrum. The cerebellum is a sort of smaller brain-like structure that sort of it attaches to the brainstem, sort of in the back there. And so this would result in Huntington's disease. It's one of the changes that occurs that results in some of the symptoms of Huntington's disease. So this is a degenerative disease that eventually will lead to death. But the early stages, people show loss of motor control. This is a nice illustration from your book showing you how some of these inhibitory synapses would work. So GABA will bind. So as long as you have two, it's a ligand-gated ion channel. So if you have two GABA molecules binding to it, there's a conformational change of this channel. The channel will open, and then chloride channels will move down their concentration gradient from outside to inside. If negative ions are moving in, this will hyperpolarize the cell. Again, the equilibrium potential for chloride is often quite close to the resting membrane potentials. The degree of hyperpolarization may not be that great, and yet it definitely has a strong inhibitory effect. So recall that peptides are chains of amino acids, and it turns out there's a huge variety of neuropeptides that are actually quite important. We're not going to go through them all because there's simply too many of them. It's almost an endless list. But I'll just name a few of these. So these are chemicals that may be used as hormones. They can act as paracrine signals. They also may act as neurotransmitter. Cholecystokinin 1, CCK. It's a hormone that's found in the small intestine, so it has to do with digestion. It's also a neurotransmitter used in the CNS. It's involved in the feeling of satiety. In other words, after you have a meal, you feel satiated and you don't feel like eating more. So it's involved in quite a diverse number of things like that. Another one is substance P. This mediates sensations of pain. Also within this group of polypeptides would be endogenous opioids. So you have receptors that are opioid receptors in your body, in the central nervous system in particular. These receptors bind to certain drugs such as opium and morphine, and this results in pain relief. So we first knew historically about these exogenous agents, in other words, agents from outside of the body that would have these effects. And then later on, scientists found the endogenous opioids. So these endogenous opioids are polypeptides. So they're small chains of amino acids produced by the brain. They can also be produced by the pituitary gland. Opioids are produce euphoria, so they may mediate reward pathways. And they may be related to a feeling of well-being after exercise. Some opioid receptors are blocked by naloxone. So it turns out there are a lot of deaths by overdose of opioids because we have opioid receptors within the brainstem in regions that generate the rhythm for breathing. So those regions will essentially stop generating a rhythm when activated by opioids. But naloxone is an antagonist. In other words, it will block the effects of opioids on these receptors. And so it's turned out to be a very useful blocker or drug in the case of overdose. So people that have overdosed on, say, opium or morphine or some other opiate drug can be saved through the injection of naloxone. Okay, another class of drugs are the endocannabinoids. These are neurotransmitters that bind to the same receptors that the active ingredient marijuana, THC, binds to. So endocannabinoids are a little bit interesting. They're not peptides or amino acids or anything. They're short fatty acids produced in the dendrites and cell bodies, and they're released directly from the cell. So in other words, they're not held in vesicles. And actually, this makes logical sense. Remember that short fatty acids, they do have a carboxyl group at one end usually, but they have a long hydrocarbon chain. So they tend to be lipophilic. They're relatively small, and so they can often pass through a plasma membrane without any trouble by simple diffusion. So it's not really possible to hold them in a vesicle for release. So they're essentially just synthesized and then released out immediately. These are very interesting also in the sense that they're used as retrograde neurotransmitters. So normally transmission goes from presynaptic neuron to postsynaptic neuron. However, there are a number of retrograde signals that go from the postsynaptic neuron back to the presynaptic neuron. And they'll indicate things to the presynaptic terminal, such as, regarding the amount of transmitter release and so forth. So in this case, these endocannabinoids may be released from the postsynaptic cell. They'll diffuse back to the presynaptic terminal, and they'll inhibit further transmitter release from the presynaptic neuron. So this is essentially a negative feedback mechanism that prevents the postsynaptic neuron from being overexcited by lots of transmitter release from the presynaptic. So endocannabinoids are really interesting in the sense that they're involved in hedonic pleasure. So this is the pleasure, hedonism has to do with pleasure. And so the types of pleasure you normally experience in life would be things like eating, drinking, sex, exercise. And so endocannabinoids have been implicated in all those different types of pleasures. Endocannabinoids can enhance learning and memory. So that's good. And that makes sense, again, because often learning and memory are actually tied to reward systems. So in other words, if you do something like, you prepare a meal and it turns out delicious. Well, you want to remember that experience, right? How you prepared the meal. So, it sort of goes together that pleasurable things or things you want to, or that are salient, in other words, they stick out in life, and you want to remember what you did to arrive at that pleasurable experience. Same thing goes for drinking alcohol, sex, and exercise. These can also obviously involve things like learning and memory, remembering, you know, well, let's use the example of sex. Like, well, sex would be something like remembering, what you did to attract someone of the opposite sex. Okay, I'm going to stop there before I get in trouble. Now, marijuana use actually impairs learning and memory. And the reason for this is the action of THCs when it's an exogenous chemical. In other words, it's not the natural chemical from within the body. It's going to just, activate these neural circuits in an uncontrolled manner. And so it really actually has the opposite effect. It's going to inhibit learning and memory. So marijuana has a number of negative effects. It can impair learning and memory in the short term. There's evidence that it can actually lead to longer term memory problems, even after a person stopped, taking in marijuana for a while. Now, in developing children and young people, by young people I mean roughly under, say, the age of 25, it's been shown to decrease IQ scores, and it's been shown to decrease scholastic achievement. And again, this may be due to the fact that these endocannabinoids really are very important for learning and memory. So they're naturally part of how you'd make associations between doing something right and getting the pleasure. For hedonic pleasure could really even be something as simple as doing well on an exam or something like that. So again, you learn what steps you did to do well in the exam in terms of study skills and so forth. So marijuana is going to sort of disrupt these circuits and this type of learning that you're doing. So it's been known for quite a while that there's some really interesting neurotransmitters out there. So nitric oxide is one of them. It's a gas. It's produced by neurons in the CNS and as well as neurons in the PNS from the amino acid L-arginine. So it's a gas, and we've talked about this, that anything that's a gas, like, say, carbon dioxide or oxygen, well, it can diffuse straight through plasma membrane very easily. And so that's exactly what happens. As soon as the gas is produced, it diffuses across the presynaptic axon plasma membrane. So it can't be held in vesicles because it's a gas. There's no way to hold it and store it. It diffuses into the target cell and activates the production of cyclic GMP, typically, which is a type of second messenger. It can cause things like in the peripheral nervous system, it'll lead to blood vessel dilation. Can also help kill bacteria. And it may also act as a retrograde neurotransmitter. In the peripheral nervous system, nitric oxide is secreted by autonomic neurons onto cells of the digestive tract, respiratory passageways, and in the penis, and it causes muscle relaxation. So nitric oxide is responsible for an erection, and the drug Viagra works by increasing NO, or nitric oxide, release. Carbon monoxide is another very interesting neurotransmitter. Carbon monoxide is actually very toxic. It can kill you. You can have carbon monoxide poisoning, and that will lead to death because it will compete with oxygen at hemoglobin within your blood. But in small quantities, it's another gas that's used as a neurotransmitter within your nervous system. So it's derived from the conversion of heme to biliverdin. It activates the production of cyclic GMP in target cells, and it's used in places like the olfactory epithelium. So this is involved in smelling, and in the cerebellum. Again, this is the sort of smaller brain-like structure that's involved in motor coordination. ATP and adenosine are also neurotransmitters. These can be used as co-transmitters. So often in a vesicle, you'll have a transmitter, a neurotransmitter, and then maybe another transmitter or even a third neurotransmitter. They're all released together. ATP and adenosine are classified as purines. So, and they bind to purinergic receptors. So certain receptors are for ATP, and for example, P1, and P2 receptors are for adenosine. One example of co-transmission would be released with norepinephrine. Norepinephrine stimulates vessel constriction in many of the tissues of the body, such as adipose tissue. So at the same time, these are released together to help also with the blood vessel constriction. These ATP and adenosine could also be released with acetylcholine to stimulate intestinal contraction. ATP and adenosine can be released by non-neuronal cells also, acting in a paracrine fashion. They're involved in blood clotting, taste, and pain. I don't really expect you to memorize absolutely everything here, but this is sort of just an interesting table just to get some general idea of where these things lie. So, for example, we talked about serotonin, histamine. These would be amines. A type of amine would be the catecholamines. These are ones that are derived from tyrosine. That would include dopamine, epinephrine, which is a hormone. I believe it's also a neurotransmitter. Norepinephrine, which is a hormone and a neurotransmitter. Acetylcholine is a little bit interesting. You see how it's all alone? It's a very ancient neurotransmitter, and it's a choline derivative. It's sort of on its own. It's a little bit unique. There are amino acids that act as neurotransmitters. We talked about GABA, for example. Glutamate, which is the same as glutamic acid. Those are identical. And we talked about glycine. So those are amino acids. We didn't really talk about aspartic acid. That's another one. And then, the list is almost endless in terms of polypeptides. Remember, a polypeptide is a chain of a few amino acids. Glucagon is a hormone. It's involved in controlling glucose levels in the blood. We've talked about it in the past. Insulin's another one. I just mentioned substance P that's involved in pain, and so forth. Let's jump down here to okay. We talked about this one, vasopressin, which is also ADH, antidiuretic hormone. We just talked about cholecystokinin, CCK. Now there's also these really interesting endocannabinoids, which are actually lipids. Remember, there's short chains of fatty acids. For gases, we have nitric oxide, and what else do we have? Carbon monoxide, and then for the purines, we have things like adenosine and ATP. Okay, we're going to end this chapter on synaptic integration. So within your nervous system, there are very complex neural pathways. As I've mentioned in the past, axons have collateral branches, so they'll bifurcate, meaning they'll split in two, and then those collaterals may then split again. And so this means that a presynaptic neuron can potentially form synapses with several or maybe even hundreds of postsynaptic neurons. So that's this idea of divergence, which you can see in this picture here. Here's a presynaptic neuron. And actually, this isn't very realistic. Usually you have a bifurcation. Here they show a trifurcation, which never really happens, or would be unusual at least. But you can see it splitting into three, and then you have these parallel axon collaterals, and they're going to go to different neurons. So that would be divergence. You can also have convergence. So you can have several presynaptic neurons, up to 1,000. They can synapse on one postsynaptic neuron. So for example, here we have 3 presynaptic neurons that all converge on one postsynaptic neuron. So that would be an example of convergence. Okay, I've also talked a little bit about summation in the past. So one thing that a postsynaptic neuron does is it will take all the synaptic input that it's getting and it'll summate it. And another word for summation is integration. It'll take all the synaptic input and integrate it or summate it. And this occurs at the spike initiating zone, which is typically around the axon hillock or the initial segment of a neuron. And so, for example, you can have spatial summation. This occurs when you have the convergence. So you have signals from different neurons coming on to a single postsynaptic neuron. And you can have some of these might be excitatory, some might be inhibitory. And so they're all going to have a local effect of depolarizing the dendrite that they contact, for example. Then there'll be a graded transmission of the depolarization to the spike initiating zone. And then there'll be a summation of all these inputs. And if the summated inputs drive the membrane potential to the spike threshold, then you'll actually get an action potential occurring. If they're not that strong, then you won't get anything. So in other words, the signal will die out and you won't have an action potential. So there's a sort of a decision made every time an action potential is generated. Is the summation great enough? Is it supra-threshold, meaning is it above threshold? And should the postsynaptic cell generate an action potential? So spatial summation, again, is convergence onto different dendrites of a single postsynaptic neuron. Now, as I mentioned before, a single neuron is always receiving a barrage of, particularly in the central nervous system, we're receiving a barrage of EPSBs and IPSBs. And these things die out. They don't stay, up forever. And so, if they come in the right sequence, in other words, if they occur near the same time, nearly simultaneously, then these signals, the EPSBs and IPSBs, will sum together, and then they may again lead, if they're big enough, they'll lead to a suprathreshold stimulus at the spike initiating zone, which will then lead to the generation of an action potential. This is an illustration from your book showing this point. So, for example, we have a neuron here, postsynaptic neuron. It receives input from neuron number 2, for example, and neuron number 1 here. Now, if you just stimulate one of these neurons, you might get an EPSB, but it's below spike threshold, which is negative 55 millivolts. Now, you can have spatial summation. In other words, if you have two neurons being active, presynaptic neurons being active at the same time, action potentials travel down. They both release neurotransmitter. They might depolarize different dendrites. From each dendrite towards the spike initiating zone, you'll have an attenuation of the voltage signal. But if they attenuate, but they're still above threshold, in other words, if they're supra-threshold at the spike initiating zone, then you may get the generation of an action potential. So that's one way of doing it, of getting an action potential, is through spatial summation. The other possibility is perhaps you'll have a single neuron that releases neurotransmitter, but then there's another action potential that follows right behind the first one, and you may get temporal summation, in which they add the second EPSB essentially rides on top of the first one, and then that leads to the membrane potential reaching the spike threshold, and you can get a spike there also. So of course, normally both of these things are going on at the same time. You have both spatial and temporal summation, and they both contribute to this summation or integration of input at the level of a neuron. We're going to skip the section on synaptic plasticity. So we've already talked a little bit about why synaptic inhibition is so important. For example, with the example of strychnine, when you block synaptic inhibitory synaptic input in the spinal cord, this could lead to paralysis and things like that. If you block, so the brainstem, so the diaphragm is innervated by motor neurons that come from the brainstem. If you block inhibition via glycinergic synapses there in the brainstem, then that can lead to a kind of paralysis, which leads to asphyxiation. It's a paralysis in which the motor neurons go to the diaphragm, which is simply generating action potential after action potential, and therefore the person can never, they breathe in and that's it. They can never breathe out. So synaptic inhibition is extremely important. It's produced by inhibitory neurotransmitters such as glycine, which is predominant in the spinal cord, but you'll also have GABAergic inhibition there. And there's GABA in the brain, but of course there's also some glycinergic synapses in the brain also. So synaptic inhibition will hyperpolarize the postsynaptic neuron. So that's an IPSB, an inhibitory postsynaptic potential, and to make it less likely that the membrane potential of the postsynaptic neuron will reach spike threshold at the axon hillock. This is just an illustration from your book of how this might work. Imagine that we have inhibitory neurotransmitter released by neuron 1. So we have an IPSB. And normally the cell's at negative 70 millivolts. Now it's at negative 80 millivolts. Now we have an EPSB. Now, normally if this EPSB had occurred all by itself, we would have reached spike threshold at the spike initiating zone, which is, again, near the axon helican initial segment of the neuron, of the postsynaptic neuron. But now, because we're starting from a lower membrane potential, more hyperpolarized, it doesn't reach spike threshold. So this is a nice example of summation, where if you have inhibition and excitation at the same time, you may not reach spike threshold. Okay, towards the beginning of this chapter, we talked about the fact that it's possible to have an axo-axonic synapse. In other words, you may have a situation in which a neuron synapses on the axon of a second neuron, and typically it's not just the midpoint of the axon or something like that. That would be very unusual. It's usually the presynaptic terminal, the synaptic bouton or the synaptic bouton, and there it can inhibit the release of excitatory neurotransmitter from the second neuron. So let me try and draw a little picture of what this looks like. So you have a neuron, and you have another neuron. Okay, and you have a postsynaptic neuron over here. So these are the synaptic vesicles. So this neuron here, let me see if I can get back to the laser pointer here. So this neuron here makes an axo-axonic synapse. Now this neuron here is going to release neurotransmitter, which will diffuse across the synaptic cleft and activate the postsynaptic neuron. Now, if there's a couple possibilities. One is that this other neuron will release neurotransmitters such as GABA or glycine that could then inhibit or hyperpolarize this this synaptic bouton or axon terminal. Therefore, when an action potential travels down its axon and invades this axon terminal, because it's more hyperpolarized, then there'll be less activation of voltage-gated calcium channels, therefore less influx of calcium, therefore less release of neurotransmitter. So that's one possibility. Your book, and that's probably the more common example. Your book actually points out another possibility, which is quite interesting, which is that you have some instances in which the neuron that forms the axo-axonic synapse is going to release an excitatory neurotransmitter. Now, this seems counterintuitive, but that can actually depolarize this neuron that's going to release the neurotransmitter onto the postsynaptic neuron. And by depolarizing it a bit for a prolonged period of time, it can inactivate calcium channels. In other words, the calcium channels are open, but then they'll close due to inactivation. We've talked about sodium channeling activation with respect to the action potential. So if the calcium channels are, at least, or some of them out of a population of calcium channels on the presynaptic axon terminal are inactivated, then when an action potential travels down this axon and invades the cell, there'll just simply be less calcium flux through, because inactivation closes the calcium channels. And therefore, again, this will lead to less transmitter being released by the presynaptic terminal onto the postsynaptic cell and less of an effect. So it turns out there are endogenous opioids that reduce pain through this mechanism. So that's very interesting. You can also see the inhibition of release of substance P, which is involved in the promotion of pain transmission. So these are some interesting ways in which presynaptic inhibition can occur and can affect neural transmission. Okay, we'll end there on this slide.